Hi Sylvain.
Hi.
Can you please tell us a little bit about yourself and then about the poster that you presented here?
Yeah, so I'm Sylvain Slavy. I'm a postdoctoral researcher from the University of Le Havre in France. I do my research in ecotoxicology, and as part of the RedPol project, I developed an in vitro assay, a new assay to detect estrogen-like endocrine disrupting chemicals in the environment. The context is EDCs. These compounds are very problematic in the environment because they threaten biodiversity and they are problematic to human health. And the current solutions are only focused on human features, so they don't represent all features we can find in the living, in the biodiversity. So we developed a new test, a new in vitro test, a non-animal based assay, which use a cell line which are modified, and we give the capacity to these cells to express estrogenic receptors identified first in a fish. They are called ESR1, 2A or 2B. And we also give the capacity to these cells to express a reporter gene, which will be expressed only if a substance, a compound, bind to the estrogenic receptors. So then we can use these cells, these modified cells, to track, to find EDCs. And we can expose them to various kinds of matrices, such as compounds, alone or in mixtures, or environmental samples. And thanks to the modifications, if the cells are exposed to EDCs, they will emit light. So at the end of the experiment, if the cells emit light, that means they were exposed to a problematic compound. Yeah.
Thank you. Thank you. Hi Anaïd, can you please tell us a little bit about you and about the poster that you're presenting here?
Sure. I'm from the University of Caen and University of Le Havre in France, and I'm working on developing tools to use natural color change in animals to use as a proxy for the assessment of endocrine disruptors. So, well, when you think at animal color change, you may think that cephalopods, because color change is quite spectacular, but actually many other animals like crabs or shrimps can change color also. It's less spectacular anyway. When you put them in dark or light environments, they change color quite quickly in about minutes to hours. So we try to use these different models, either by putting them in a specific environment to induce color change, and we already evidenced that color change abilities were altered by exposition to pollutants such as endocrine disruptors.
And what would be the consequence of this interference of pollutants with the color change? What would happen with the animals if they change color in a different way?
Yeah. Basically, the animals use color change for a range of purposes, including protections against UV, camouflage, or any form of communication. So all these biological functions will be altered to some extent. So we can imagine alteration of their survival on the long range.
Thank you. Hi Caroline, can you please tell us a little bit about yourself and about the poster you're presenting today?
Yes, so my name is Caroline Arcanjo. I'm working at Le Havre University. I'm a postdoc researcher, and I am working on very small crustaceans, which is called the copepods here. And so we use them to perform behavioral assays that we developed in the lab. So we use an observation chamber, which was initially developed for small fish larvae. So we adapt this tool for our copepods that are at least twice smaller than fish larvae, and also not so pigmented. So it was quite hard. But we managed to adapt the tool. So then we used bioassay for tracking the swimming behavior of copepod and to see if the pollutants in the project, which are suspected to be endocrine disrupting compounds, have an effect or not on the swimming behavior of this little crustacean.
Thank you.
Thank you.
Right, so hi Bidemi.
Hello.
Can you please tell us a little bit about yourself and about the poster that you're presenting today?
Yes, thank you. So my name is Bidemi. I'm a PhD student at the University of Portsmouth, and my poster is on how plastic additives affect the behavior of amphipods, especially river shrimps. They're really tiny animals like this. So basically we expose them to different concentrations, environmentally relevant concentrations of plastic additives, and that's DHP and DBP. I have the name somewhere here. Yes, DHP and DBP. So they are phthalates and they are very present in plastics and products of plastics in our everyday items, and it's been known that they have really toxic effects on animals, but there's very little research on how they affect amphipods. So that's the essence of this research, to know what they do to them in terms of their behavior. So basically the experimental setup is done such a way that we are able to track how the animals swim over a certain period of time, and this is the Daniel Vision machine. It tracks the animal in form of infrared, and it takes record of how the animals move in a period of light and dark phase for a total of 8 minutes. So this is the results of the experiment. So this is the dark phase, the light phase, dark phase, light phase, and this was done for a period of 14 days. So I did the first tracking on day 7 and the other tracking on day 14. And from what we can see here, the animals tend to move more in the light, which is what we expect. And then when the time goes into the dark phase, they reduce the speed of their movement. So how do we know if a compound has an effect? So basically, if there's a compound effect, the animals that would ordinarily move more in light will tend to move less when the light comes on. So that's how we are able to hypothesize that yes, indeed, there's a compound effect on these shrimps over a period of time. And from what we can see here, we had more effects in DBP compared to DHP. And this is an ongoing work. We're still trying to optimize the entire procedure of carrying out the research. And from what we have here, we can obviously see that there's a toxic effect of this DHP and DBP compound. Even though they've been banned, they are still very prevalent in the environment, and more regulations need to be done to limit the presence of these compounds into our aquatic environment. Thank you very much.
Thank you.
I'm not sure I can remember.
Just quickly explain what's in there. I'm sorry, I don't know. So if you just wrap up in 2 minutes, what's that?
Hi, my name is Dr. Sam Robson. I'm a reader in genomics and bioinformatics at the University of Portsmouth. I'm here showing some of the equipment that we use in my lab for doing DNA sequencing, which we use for a variety of different reasons. One of those being working within the RedPol project, looking at the effect of endocrine disruptors on gene expression in a variety of amphipods. What we've got here is a collection of some of the machinery that we use within my lab. This is our bento lab, which is essentially an in situ lab. It's a lab in a box that we can take out into the field, and we can perform standard molecular biology techniques such as DNA extraction and the library preparation for the DNA sequencing that we perform. So we're in the process at the moment of trying to use this to take out into the field with us for doing, in particular, microbial analyses, looking at barcoding what samples might be available, might be around within water samples, for instance, or in soil samples collected from environmental locations. Over here we have, this is the MinION sequencer. which is a DNA sequencer available from Oxford Nanopore Technologies. We use this technology because it's very portable. As you can see, it's very small, about the size of a stapler, but the machine itself is able to generate about 30 to 50 gigabases of data, which is technically enough to cover the whole human genome over 10 times. And we're able to take that out and about with us and do these kinds of sequencing experiments out in the field, as well as using the technology for a lot of the research that we do within my lab.